name is Mateusz Chiliński, and today I'll be presenting video presentation of a poster entitled Consensus Based Identification and Comparative Analysis of Structural Variants by Long and Short Read Sequencing Technologies in Selected Human Families. What we did first in our work, we've done independently 12 SV callings, as you can see, with tools like Breakdancer, BreakSec, Sinnovator, Deli, GenomeStripe, Lumpy, Manta, NovaBreak, Pindel, Svelter, TARDIS, and FHAMK. After we have done all of those callings, we decided to create an algorithm for actually merging those callings to obtain a high quality data set with all of the tools inside. You can see the algorithm pipeline on this slide. As you can see, the input to the tool is independent call from all of the other algorithms that call the structural variants. Then we go to the pre-processing for the tool, because PCF format is pretty standard. However, the output that is provided into the info field varies from tool to tool, so we need to unify it for the further processing. After that step is done, we try to find structural variants that are close to each other in terms of the position, length, and type from various tools. We call those found SVs as candidates, and then we simply take the candidates and we check if the majority of those are exactly the same in terms of the position and length. And if they are exactly the same, then we can simply use one of them to create consensus, because it's a pretty solid and accurate reflection of the structural variant. However, if majority of the candidates differ, we are using the pre-trained neural network model to establish the consensus. After that, genotype is established based on the voting algorithm, and we get one unified VCF file with all the SVs with the names of the algorithms that were used for the establishment of this particular structural variant in the info field. Now I'll show some results of our consensus SV. We have compared ourselves to a cold set of structural variants published by Jason et al. In 2019, and as you can see, we have obtained around 85% of the accuracy when we compared our results to the gold set. We covered 85% of them, while when we did the opposite comparison, the data set that was created by this gold standard publication covered 66% of our set. Then, after doing the whole pipeline for the Illumina, we decided to compare the results with Oxford Nanopore as well. We have used three structural variant colors. In case of Oxford Nanopore, as you know, it's the technology for obtaining long reads. We don't need to use that many structural variant colors because it's simple technology that is in favor of detecting structural variants. So we use Sniffles, SVIM, and Qt SV. We also applied our consensus SV algorithm to merge them together. We have taken the trios from 1000 Genomes project. We took actually only the daughters from those, and we ran the whole pipeline on the Oxford Nanopore technology, as I mentioned, and Illumina one, and the results are presented in this table. As you can see, in case of Illumina, we obtained around 4000 structural variants from the whole pipeline, and in case of Oxford Nanopore, it was around 24,000 structural variants. So it's a great increase in numbers, which also only supports the fact that Oxford Nanopore detects much more structural variants than short read technologies. In case of the length distribution, as you can see, we put them on three charts here. You can see that Oxford Nanopore outperforms Illumina in almost all the bins that we created here, and well, it's detecting much more structural variants, especially the smaller ones, which is quite obvious because, well, when we have much longer reads, then it's easier to detect in one read the structural variant because something is missing in comparison to the reference genome. What we want to do next, we want to create the fully automatized pipeline for the structural variant processing from the raw FastQ data to the final consensus VCF file, so it's all automized and wrapped into a Docker image, so it's really easy to install and use. And for Illumina, we'll incorporate 10 SB colors, mostly because of the license issues. We won't be using 12 as in this study. And for Oxford Nanopore, we'll use the 3 structural variant colors that were also featured in this study. That's all. Thank you for your attention.